Hello, my name is Robert Binder, and I'm an associate professor in the Department of Immunology. The focus of my lab is at the interface of cancer and the immune system. We have uncovered a pathway through which the immune system is able to detect and respond to cancer. Heat shock proteins are present in all cells. They are intracellular proteins, and they are kept this way for very important reasons. In cancer cells, because of dysregulation of many of these pathways and leaky membranes, these heat shock proteins can be released into the extracellular environment. In this space, they are able to engage a receptor that we discovered in my laboratory called CD91, which is expressed on dendritic cells. When heat shock proteins engage CD91, it triggers events within the dendritic cell that leads to priming of T cells and activation of NK cells, which are anti-tumor. And these cells can now eliminate or eradicate the cancer cells that are present in tissues. We have invoked this pathway in cancer immunosurveillance. So here you can see when you have a growing mass of tumor cells, these will release heat shock proteins that can be recognized and taken up by dendritic cells through the mechanism that I just described. If you interrupt this interaction between heat shock proteins and CD91, you do not get immune responses, and the tumors remain blind to the immune system. What we've also been able to do for many years now is to use heat shock proteins as cancer vaccines. Essentially, you can treat mice with heat shock proteins that are derived from the tumor, and if you challenge these mice with tumor cells, the tumors fail to grow, or they grow much slower in the treated mice. And we've been doing this in humans also. If we have patient tumors, we can extract heat shock proteins from these tumors and administer the purified heat shock proteins back into patients with the idea that the immune response will be activated in this patient, leading to eradication of the tumor that has been left behind by the surgeon or metastases that may occur in distal tissues. And we have several clinical trials that are ongoing using this mechanism. There are many major techniques that are being used in my laboratory, and I will invite you to write to me to discuss rotation projects that are available in the lab. The lab is comprised of a group of very dedicated and passionate individuals who are all aspiring to make major discoveries, and I invite you to join our group. Thank you.